Hello everybody, I'm Yogan and I'm representing protein core Cayman in chemical. Today I'm going to talk about the production and characterization of N and C terminal selenocysteine proteins. Selenocysteine is the selenium-containing analog of the cysteine. It is the 21st proteogenic amino acid available in biological kingdom. And remarkably, while some organisms like higher plant and fungi use the readily available sulfur to express cysteine homologs, mammals, fishes, birds, nematodes, and the bacteria still maintain the energetically costly and inefficient process of selenocysteine expression. Here we want to show the efficient production of N and C terminal selenocysteine protein in the E. coli cell and their characterization. There are 25 selenocysteine proteins in human. Here we assess their amino acid sequences from Uniprot database, and a phylogenetic tree was built to know the relatedness. The location of the selenocysteine residue contained in each of the proteins are tabulated considering the selenocysteine at N and C terminus or around the middle of the sequence. Some of the proteins consist of more than one selenocysteine residue. Most remarkably, the selenoprotein cell P has 10 of those and shown in green. The clans of the glutathione peroxidases, iodo-thioredoxin deiodinases, and thioredoxin reductases are shown in orange, pink, and blue respectively. Here we focus on N and C terminus selenoprotein considering the representative example of GPX4 and rat thioredoxin reductases respectively. The right panel showed the crystal structure of GPX4 and rat thioredoxin reductases showing the selenocysteine residue in a color dots. The top panel showed the overlaid crystal structure of the mutant selenocysteine 46 to the cysteine in shine and a wild type consisting selenocysteine at 46 residue in green for the GPX4. The bottom panel showed the crystal structure of rat thioredoxin reductase in which the FAD and NAD are shown in red and blue respectively. Glutathione peroxidase is an enzyme family with a peroxidase activity whose main biological role is to protect an organism from oxidative damage. The biochemical function of glutathione peroxidase is to reduce the lipid hydroperoxides to their corresponding alcohol and to reduce the free hydrogen peroxide to water. GPX4 functions downstream of the glutathione to repair lipid peroxidation and is the key enzyme for the ferroapoptosis. There are 8 GPX proteins in human, however only 5 GPX1, GPX2, GPX3, GPX4 and GPX6 constitutes of the selenocysteine residue. The selenocysteine are located at N terminal. The portion of the amino acid alignment of those proteins is shown and the Sec-residue is highlighted in red. Our study focused on GPX4 and highlighted in green in which the Sec-residue is at 46th residue. Since the heterogeneous expression of the Sec-incorporated GPX4 in E. coli is challenging, we use an engineered E. coli host C321 delta express transforming 3 extraneous plasmid T7 RNA engineering tRNA and cell a GPX4 combination. We obtain soluble GPX4 by this way. With our in-house engineered expression system, soluble protein was purified. MS analysis and fragmentation profile of the protein was analyzed to confirm the incorporated selenocysteine. The selenocysteine containing peptide showed the carboamidomethyl plus 57 modification for cis-2 CAMP-SEC plus 105 with the observed ion M over Z of 844.38 as shown in the peptide fragmentation in table B. We also confirmed the GPX4 activity using Cayman's glutathione peroxidase assay kit. The activity of the purified protein in gray was measured and compared with the background in blue and the positive control in green as shown in C. The specific activity of the purified protein was measured using the serially diluted GPX4 protein in the same assay kit, and a unit activity was defined accordingly as shown in figure D. Thioredoxin family proteins comprise of the thioredoxin, abbreviated as TRX, and the selenocysteine containing thioredoxin reductases, abbreviated as TRXR, either in the cytoplasm, mitochondria, and in the nucleus. The catalytic activity of the TRX and the TRXR1 during the oxidative process play a role in immune function and cell apoptosis. There are 3 thioredoxin reductases in human. TRXR1 in human and the rat are identical, and all the homologs have penultimate Sec-residue at the C-terminus region. The portion of the selenocysteine incorporated alignment of those proteins is shown and highlighted in red. We focus on rat thioredoxin reductases R1 and highlighted in green, in which the selenocysteine residue is at the 490th position. Being a larger protein, the heterologous expression of the selenocysteine incorporated protein is difficult to obtain. The expression of rat thyroxine reductase R1 was done in engineered E. coli host C321XP using tricystonic expression placement consisting of cell A, cell B, and rat thyroxine reductase R1 with the various modification which was originally described by Elias. With our modified method of heterologous expression and purification, soluble protein was obtained. Preliminary characterization of the protein was done spectrophotometrically by detecting the spectral characteristics of FAD, which showed the typical absorption maxima at 4s63 nanometer. Then we did the MS measurement and analyzed the fragmentation profile of selenocysteine containing TRXR1 peptide. The Sec-containing peptide showed the carboamidomethyl plus 57 modification for cis to camp selenocysteine of plus 105 with the observed ion M/G of 629.72. Then the activity of the protein was determined using Cayman's thioredoxin reductase colorimetric assay kit using TRXR1 inhibitor sodium orothiomalate ATM as a negative control. Here we describe the details of our selenoproteins and the related assay kits. For GP-Expo, glutathione peroxidase assay kit was used, and for the thioredoxin reductase colorimetric assay kit was used. Under the umbrella of Cayman's bioanalysis and assay development services, Protein Core provides the service from in silico engineering of genes to the protein production and characterization. In addition, Cayman's Protein Core also serves for the native protein isolation, labeling, PTM mapping, and we have also access of in-house crystallography.